I am really impressed at how many of you are here tonight. I don't know how, after teaching all day, that you had the energy to drive all the way into Boston and in order to attend this. So I really, first of all, just admire you for what you do every day, and hope that what you hear today is worth the effort that you put into being here. Certainly, you know, from my experience and many of my friends, as high school teachers, you have such a profound effect on the careers of young people, and so I think these kind of things that you can enrich the teaching experience through even anecdotal stories certainly helps a lot. So today I would like to tell you a little bit about what's known about the etiology of breast cancer, and then tell you about some studies, or just give you an idea of the kind of studies that are going on in our lab and others in order to further our understanding about the molecular pathogenesis of cancer. So I broke the rule for all PhDs that Hal told you about. I have blue background, not white, but it's kind of an aqua blue, and real doctors never use anything but the basic color. So I think this is somewhat acceptable. So just a little bit of background. I actually left high school with an interest in becoming a math teacher. And that was because my math teacher in high school was absolutely fabulous. And she got me so excited about math that I wanted to be able to, inspire young students for the future to be a math teacher. But then luckily I had a fabulous biology teacher for my general intro to bio, which I took as one of my core science classes. And this person really turned me on to science. It was when the genetic code, this was a long time ago, this is going to age me. It was when the genetic code was first being derived, and protein translation was just starting to be understood, and how triplet coded for amino acid and the involvement of ribosomes. And I was so excited by that I actually changed major to a biology major, and I'm certainly glad that I did. And then I had a personal experience in high school where I wanted to learn more about cancer, and took an independent study while I was at Northwestern. And through that independent study, I read a lot of research papers, or at least I tried to read. I didn't really read them now that I think back on it. And it exposed me to the whole arena of scientific discovery and design and interpretation of experiments. And I basically got hooked by that experience. And so I decided, instead of being a biology teacher, to actually try to do research as well as teach. So I went and did a summer research program at Jackson Laboratory in Bar Harbor, Maine, just to get an idea about whether I liked research or not. And then I've kind of been hooked ever since. So I guess that's my reason for feeling like teachers can have such a profound influence. So this slide is really superfluous after Hal's talk, which was just a perfect backdrop to mine. But I just wanted to reiterate that breast cancer is the second leading cause of death among women ages 35 to 54. And this fact is partly what motivates me to come to work every day. And clearly, further, as Hal pointed out, further research is needed to identify factors that cause breast cancer and to improve the detection and treatment of this disease. So I'm going to tell you about kind of things that we know now about what causes breast cancer, and what's being done to learn more. So feel free to ask questions. You're not going to be able to communicate this to anybody else if you don't understand what I'm saying, and so we tend to forget, because of the jargon we use, that some of it is not really intelligible to even people with good scientific backgrounds or good biology backgrounds. So please ask questions. So basically, the challenge is to identify the Achilles heel of tumor cells. And I think this is a good analogy, because Achilles was resistant to lots of insults because he was dipped into because his mother dipped him into this fountain that made him, or this pond, whatever, that made him resistant to all kinds of attacks. And tumor cells clearly have evolved to be resistant. They've escaped all the normal checkpoints and controls that normally would limit their survival and their proliferation. But there are Achilles heels of tumor cells, and the real challenge is to identify the unique sensitivity of tumor cells so we can target those for therapeutic intervention, because that's going to be the best way in which we can selectively kill tumor cells without affecting normal cells. And that's really the goal. However, it's a really big challenge because there's enormous heterogeneity in tumors, not only in different types of tumors, but within individual types of tumors. Genetic instability allows this tumor cells to change in order to escape from the therapies. And there's a very complex interaction between the tumor cells and the cells in their environment that really complicate the task of identifying these sensitivities. So if you read Time magazine, you'll know that, and hearing Hal's talk, that what we now realize is that a major frontier in cancer therapy is really designer therapy. So like Hal said, we want to identify what are the specific events that are driving individual tumors, and then have therapies for a whole variety of different drivers in order to be able to specifically eliminate tumor cells based on their specific, the specific set of alterations that took place in individual tumors. Okay, so as Hal mentioned, this is really the first glimpse that anyone gets on what's the nature of any specific tumor. So the tumor is excised by a surgeon, and then the pathologist takes a look at it. But this doesn't really tell you anything about what caused that tumor or what are the cellular events that are driving it, what are the mutations. And so in order to develop therapies that are designed to target events that drive this, we have to have a way of basically understanding what events led to the development of that tumor, and what are the specific cellular proteins that are required in order for this cell to display its abnormal growth properties. So we want to understand what processes, proteins, genes are responsible for the events associated with the initiation and the progression of the tumor. Now, all breast tumors don't have the same appearance. So these are just the architectural features or the histology of a variety of different grades and stages of breast tumors. So here you have the normal little glandular structures. And in this case, one of the early stages of disease is where you just have proliferation of the cells that form the outside of these little glandular structures, which I'll show you more about later. And then later stages of the disease, those proliferating cells are actually able to fill the hollow luminal space. But then you have different grades of this carcinoma in situ, or non-invasive carcinoma. So you can see here, these cells are all relatively uniform size. The nucleus has relatively uniform morphology. But in higher grades, each of the individual tumor cells is much more heterogeneous, and there's considerable distortion of the architecture of the individual cell and the nucleus relative to normal cells. And then obviously when you have invasive carcinomas, there's a breakdown of the membrane, the basement membrane that delimits these tumors. The cells acquire ability to move into foreign places and to survive outside of their normal environment. So our lab is really interested in understanding what distinguishes these different stages of tumor progression. So for instance, in both of these early stages, the cells are able to escape the normal growth control and they hyperproliferate. What distinguishes a tumor in which the cells fill that space, what's called a luminal space, the lumen of the gland, from those that don't? And what are the genetic and cell biological processes that distinguish these high-grade tumors from the low-grade? And then what events allow some of the tumors to actually invade into the surrounding tumor environment? So I'm going to, just before I tell you more about that, I just want to give you a little bit of basic background into the architecture of the breast and the mammary gland and the glandular structure itself. So this is just a cross-section through a human gland. And what you see are the ductal system. You see the ductal system are like pipelines that lead out to what's called a terminal ductal lobular unit, which if you look more closely at using a scanning electron microscopy, which is one of my favorite pictures in the world. So each one of these individual units is one of the end buds of this terminal ductal lobular unit. And if you look in cross-section, say through one of the TDLUs, you see that each of these little structures has a hollow lumen. And this is just a cartoon of what each one of these structures would look like. So basically you have a layer of what are called luminal epithelial cells surrounding a hollow lumen. And it's into this lumen where milk is normally secreted in a pregnant gland, in a lactating gland. And then the milk is secreted into these end buds, and then it's transported through the pipelines out to the nipple. So this cartoon and some of these sections just illustrate in a three-dimensional way with the nature of the events that are associated with these different stages of carcinoma in situ. So in one case, the hyperplasia, the cells just proliferate within this monolayer of the gland itself, so that it keeps getting bigger, but the lumen is retained. And then in carcinoma in situ, those tumor cells are actually able to fill that luminal space. And then invasive carcinoma, the cells are able to break through this basement membrane, which is extracellular matrix proteins, and they're motile and they're able to invade. So we and many, many others are interested in understanding the specific events that are responsible for that. So as you know, one of the most important, and you heard from Hal, one of the most important aspects of the development of the tumor is that these cells acquire mutations which allow them to progress through these different stages. And different types of breast tumors contain mutations in different genes. This isn't like there isn't one set of mutations which always take place in tumor cells. I mean, in breast tumors, almost every tumor cell basically has a different combination of insults that led to the progressive changes that occur during the development of a breast tumor. And what's the nature of the mutations? There's a wide variety, as I'm sure you're aware. So first of all, there's very gross changes in the chromosomes themselves. As Hal told you, like with HER2 or NU, there's amplifications of certain regions of certain chromosomes. There's also deletions. There's very, very precise technological tools that are available now that can actually look at almost every portion of every chromosome. This is something called comparative genome hybridization, and it's actually been displaced now more recently by techniques where you array individual pieces of DNA that cover the whole genome, and you can tell exactly which small set of genes are either upregulated or mutated or amplified or deleted in cancer. But this just shows, for instance, I just wanted to show you this to show you that in any one particular tumor, there's lots of amplifications, which you see here, or deletions. So each tumor has a unique spectrum of alterations, and most tumors have multiple alterations. And then there's much more subtle mutations as well. For instance, you can just have a point mutation, which causes the protein either to have lower or greater activity than normal, or that is unregulated. So normal proteins, as you know, are normally in an off state, and then they're activated by some normal physiologic process to be on, and then there's another process that shuts them down. But many mutations cause that activity to be unregulated, so the protein is constitutively on and therefore leads to a constitutive activation of intracellular pathway. Also, some of the mutations are not in the coding sequence itself, but in the promoter sequences or the transcriptional control sequences upstream, so that it either prevents a protein from being produced or a decreased protein production. And then some of the changes that occur, and this is actually really common, are not genetic changes, but epigenetic changes. So they're not specific changes in the nucleotide sequence, but they are changes in modifications of the nucleotide sequence, which actually alter the ability of that gene to be transcribed. So the most common, you've probably heard of, is methylation of cytosines in DNA. And methylation of those prevents the gene from being transcribed, so that you no longer can make RNA from that gene. So that's equivalent to a chromosomal deletion. It can no longer be expressed. And actually, for methylations, the methylation pattern is actually replicated. So when this cell with these methylated cytosines replicate, there's a replication of that pattern. So the daughter strand is actually methylated in the same place. So it can be inherited in the sense of within that tumor. Yeah.
That just prevents RNA from
being transcribed, right?
Replication of DNA is fine.
Replication of DNA is fine. Yes, right. Good question. And then another type of epigenetic change that also affects transcription is that the histones, as you know, coat the DNA, and histones, either through methylation or deacetylation, changes the state of the chromatin, the protein DNA complexes, and that can prevent transcription as well. So normally what we know now is that histones are acetylated when the gene is active, in order to change the conformation of chromatin so that it can be actively transcribed. And so deacetylation or methylation can prevent those genes from being transcribed. And so many of the genes which we know are frequently deleted in tumors are also found to be, the genes are either methylated or there's suppression through histone methylation or deacetylation. Yeah?
Is methylation kind of like a permanent off switch?
It's reversible, but it's replicated. So you can reverse it. So for instance, if you put in an inhibitor of methylation, azacitidine is one. If you put that into cells, you'll lose the methylation, and then you'll get expression. So it's reversible, but you do get a copying of that when there's replication.
What factors cause increased methylation or deacetylation?
Well, that's a really good question.
Okay.
So there's only a very small number of methylases in the cell, and they seem to be universal. So we don't know why specific genes are methylated, and understanding that would be enormously useful, because you can imagine what you want to do in say breast cancer is what people are finding now is that BRCA1, which is mutated, genetically can cause breast cancer. There's evidence now that in other tumors that are not hereditary, that are not in patients in which there's a mutation in BRCA1, that there's actually methylation. So its activity is being shut off through methylation. If you could turn off that methylation, turn back on BRCA1, then, at least in experimental models, you can revert the tumor to a normal behavior. So that's a really active area of research, and it would be a great to figure that out. And that's one of the frontiers. Okay, so the different types of alterations in tumor cells include irreversible, heritable genetic changes, reversible but heritable epigenetic changes. And then one other thing I want to mention, which is an area that is just blossoming now. And there's very few people doing research, but there's a lot of really interesting evidence suggesting that the cells that are in the environment surrounding the tumor cells, so the fibroblasts, like the stromal cells, the fibroblasts, the endothelial cells of the blood vessels, immune cells that get recruited to the tumors, and actually the normal cells, the normal non-converted epithelium, have a profound influence on the behavior of the tumor cells. So just one scenario is that what we know in breast cancer is that once you have initial conversion of the normal epithelial cells into early stage tumor cells, they produce factors that activate or change the fibroblasts that are in the environment. Then those activated fibroblasts produce factors that then feed the tumor cells. And so the tumor cells, you have to think about a tumor almost like a tissue. It's a mixture of different cell types that have mutual interdependencies. And so some of the therapies or people are looking at therapies that actually target the microenvironmental changes, because the cells in the microenvironment they don't have genomic instability. So they're not going to be as likely to be able to escape the therapeutic approaches. Like, so for instance, angiogenesis, blood vessel infiltration. The normal blood vessels are less likely to be able to escape chemotherapy, then the tumor cells, which are so genetically unstable, they can mutate to figure out a way to be insensitive to the chemotherapy. So there's a lot of interest in targeting the microenvironment, but we need to understand more about it. We know about angiogenesis, and now there's just recently some trials that the results are just coming out, showing that by blocking a growth factor that's required for recruitment of blood vessels is actually showing efficacy in the clinic. So I think you'll be seeing more and more of that. Okay, so all these proteins that are altered, what are their functions in the cell? What's the natural biological role of these proteins that are mutated in tumors? Most of them are regulatory proteins that are involved in regulating some aspect of cellular behavior. And I could give a whole course in what's the nature of the proteins that are altered. And since I don't have time to do that, just let me just make the generalization that there's basically two different classes of proteins. So as you know, every cellular event, basically every cellular event, in order for a cellular event to take place, you have to drive the event with some kind of factor, say, or some physiologic change, but you also have to remove negative regulatory factors that normally prevent those events from taking place unless all the cellular machinery is in the right place, is properly coordinated. So some proteins promote cells to proliferate, to survive or move, and generally there's an upregulation of those proteins. And then other proteins normally say halt cell division or proliferation or motility, or they tell the cells to die. And these proteins, the ones that are normally the brakes, they're lost or underproduced in cancer, whereas those that are like the accelerator, they're driving the event, they're often overproduced or mutated so that they're constitutively on. So basically, in order to proliferate, for example, you have to stimulate the cells with growth factors, and that activates a whole program of events in the cells. And so what you see is either the mutations cause a constitutive production of growth factor, or it mutates as the cellular proteins that are required to drive that. And then others cause a loss of proteins that normally are preventing those events from occurring unless all the right events are coordinated. Sorry to try to be so generous here. Okay, so let's just think, so what I wanted to do is just take you through some of those events I talked about that are involved in the initiation and progression of breast cancer, and talk about what kind of mutations could be involved there. So here we have just a cross-section through a normal gland, and when that gland reaches a certain size, when each one of these individual units reaches a certain size, there's signals that shut off the proliferation, and that prevents aberrant proliferation. So there's proteins, let's say at least one of which is this red protein that's preventing that cell from dividing. So one of the first events that occurs when you get hyperplasia is that there's a loss. So there's a deletion or a mutation in that gene that is normally preventing the proliferation. But at the same time, you're going to have to, in order to get this hyperplasia, you're going to have also a gain of function mutation that's going to drive the proliferation of those cells. So you would need both a loss of growth suppressor and something to stimulate the proliferation as well. Now, what our labs found, and I'll tell you a little bit more about later, is that we found that when these cells proliferate, they proliferate into the center of this hollow cavity, and there's normal controls that prevent those proliferating cells from living. So basically, any cell that proliferates into that space undergoes cell death, and it's a programmed cell death, or apoptosis, which you've probably heard of. Now, in order to fill that space, that space in the cavity, the tumor cells have to acquire the ability to survive in the face of that cell death program. So they need anti-apoptotic or anti-death mutations that prevent the cell death. So that would be a second type of mutation. So you need to drive the proliferation. That's not sufficient. That might give you this hyperproliferating, but you're not going to fill the luminal space. You need a survival signal. And then But these cells are going to, these tumor cells are not motile. They're unable to migrate out of this little cocoon that they're in. And so in order to start invading into this, you have to have probably activation of a protein that's going to drive its motility, as well as loss of a protein that's normally preventing those cells from being able to escape from that little cocoon. So these are the kind of events that could be associated with the development of a breast tumor. But as you can imagine, there's any number of combinations of different proteins which can be, or genes which can be altered in order to generate this state. And that's why cancer is so difficult to cure cancer, because there's basically random mutations, and there's lots of different points in any one of the pathways that control proliferation, survival, or migration that can be altered that prevent the norm, that allow these cells to proceed to the next step.
Can I ask you a question? At
Yeah.
what point is the tumor detectable? At a hyperplasia stage or carcinoma?
Well, some of these hyperplasias can actually get pretty big. So they would be potentially palpable, or I don't actually. I'm, you know, being a PhD, I don't know what a mammographer say would indicate, because often you can't see anything, I think, on mammography unless there's some kind of calcification, or I'm not sure if totally soft tissue hyperplasia would be detectable, but they can get very big. You know, some of you may have had cysts, which are basically, a cyst is not very different than these hyperplastic, where you just get, you know, a non-tumor type fluid-filled cavity, and they get really big. So I think it depends on the size. It all depends on the size and the extent to which there's a difference in the density from, say, mammography. Okay, so how many different genes or proteins are altered in human cancers like breast cancer? And as far as breast cancer, in the early days, there were just a really small subset of genes. There were those that were identified because because of their genetic inheritance, like BRCA1 and BRCA2, and then a variety of others that were identified through actually a variety of different strategies. However, with the enhanced technology that's available now through the sequencing of the human genome, we now can look at whether any protein, any gene in the genome is altered, and using any of a variety of different approaches, either looking at specific genes and whether they're amplified or deleted, and then, as Hal mentioned, you can actually look at the RNA that's transcribed from every single gene, and you can get a profile of the RNA that's expressed in an individual tumor cell, and then you can compare. So this particular array here is one, actually I think it's one of the same ones that Hal showed. This is a set of about 50 different breast tumors, and if the genes that went up relative to kind of a normal standard are red, and the genes that went down are in green, and what you can see here to some extent is there's kind of some clustered patterns, so what are called signatures. So for instance, you see that there's certain sets of genes which are coordinately up or down regulated in different subsets. If you look down in here, you can kind of pick out the different subsets. So here's a set of tumors, all of which are these genes are down regulated, but this set, there, a lot of genes in that cluster are upregulated, and then here's, a different profile. So we think that these different profiles of RNA expression are telling us something, that there's a difference in the state of these tumors that have these different signatures. And so based on looking at the outcome of these different subsets, we know that each of these different signatures the tumors with each of these different signatures has a different outcomes. Remember those Kaplan-Meier plots. So one of them, the tumor's more likely to progress to metastasis and lead to cell death, and other ones aren't. What we don't know is what that means in terms of, we know we can make predictions based on these signatures, but we don't know what it really means in terms of what genes are responsible for these different signatures or states. Yeah?
How would the array be affected if the DNA was actually methylated?
Okay, so,
Could you even look at an array then?
okay, so if the DNA is methylated, what we're looking at here is the RNA.
Right.
So what's going to happen to an RNA, what's going to happen to the transcription of an RNA if the gene's methylated? You won't see it. So it does reflect. And what's nice about it in some ways is no matter what the change is, you're going to see no matter what caused the upregulation or downregulation, you're going to see it. It could be because the gene was deleted, or because it was methylated, or because it was the histones were methylated. So in a way, it's like a readout of the function. So if you look at this, this is just a subset of the genes that were looked at. These were the ones that fit into these different signatures. But you can see there's lots and lots of cellular proteins that are upregulated, or genes that are upregulated or downregulated in tumors. And just within breast tumors, we've got 5 different subsets. And even within this 5 different subsets, it's almost like a continuum. It's not like every one has exactly the same set of genes that are upregulated or downregulated. So how do we deal with this? How do we find out what we want to know is what is causing these different signatures or cluster patterns, and what are the genes that are driving these different states. So so hundreds of proteins show decreased or increased production in breast tumors. Each tumor varies a great deal, but some patterns are emerging where different sets of genes are found to vary together. And we'd like to correlate a specific signature of genes with subtypes of tumors, or their responsiveness to therapy, or to understand what specific genes and cellular pathways are determining that particular signature. So most importantly, we want to find out how to use this information to prevent or treat disease. Okay, so this just this is just your typical signaling. This is that Rorschach plot that Hal talked about. So these are proteins that are actually involved in regulating cell proliferation, and a large percentage of them are mutated or altered in human tumors. And basically they control different aspects of cell proliferation, cell survival, and invasion. And what really wanted to do was to figure out, of all these different proteins, which are the ones that are driving the altered state of proliferation, survival, and invasion. So would it be possible to take individual genes and put them into breast epithelial cells and see what each individual gene can do, so we can find out which ones are the master drivers and the master switches versus those that are just secondary events that are a consequence of the master driver. So we want to take these altered genes, put them into breast cells, and say which ones can profoundly change the behavior of those tumor cells. So basically what's been done in the past is that these genes are altered in mice. So for instance, if it's an upregulation of a gene, they'll make a transgenic mouse that's overproducing the gene. Or if a gene is deleted normally in breast tumors, they'll knock the gene out by homologous recombination. And then for a much simpler model, people just take cells, grow them in tissue culture plates, and then if they have altered proliferation or altered uncontrolled proliferation, they'll make these little foci, and you can pick up foci. But as you can tell, these in vitro models, which are the ones we can genetically manipulate, and we can dissect the biochemical pathways by extracting the proteins and looking at their state, this doesn't really tell you much about the biological activities that are associated with initiation and progression of breast tumors. So we wanted to develop a model that was kind of in between these. So we wanted to develop a model where the breast epithelial cells would be able to organize into structures that resembled the structure in the normal gland. And so basically we wanted to recapitulate or reconstruct a glandular unit like this in a tissue culture plate, because then we'd be able to monitor biological activities that mimic those that take place during the progression of breast tumors. So we actually developed a model that was able to do that, and it was really developed through the inspiration and guidance of a woman named Mina Bissell, who has worked for years trying to develop conditions in which the breast epithelial cells could do that. So basically what we can do is take a normal breast epithelial cell and incubate it under conditions, which I'll show you in a minute, in which the cells proliferate into these small masses, and then they're actually able, on their own, to undergo a remodeling to create a spheroid-like structure with a hollow lumen. And this is just what it looks like by bright field phase contrast imaging, and this is a cross-section showing the just staining the nuclei, so you can see these outer nuclei and, for the most part, hollow lumen. There's a couple cells in there. So what we do with this, and I'm grateful to a woman named Cassie Lynn who just wrote a report on using these approaches for Journal of the American Medical Association. So she drew the cell culture technique that we use very, very nicely. So basically what we do is we take these what are called 8-well tissue culture slides, and we take the breast epithelial cells and put them on top of a gel of basement membrane proteins. So if you concentrate collagen, laminin, and other extracellular matrix proteins that are part of the normal basement membrane or basal lamina, they'll make a gel. So you put this gel down, just a little layer of this gel, and then you put single breast epithelial cells in there, and then we overlay it with a very dilute suspension of those basement membrane proteins, so a 2% suspension relative to this 100%. And what happens is that this basement membrane gel allows the cells to actually organize into structures like this. So instead of having a monolayer, we've got these structures. And what happens when we culture them like this is that they undergo this process of lumen formation. And the way in which they do it is that after 7 days in culture like this, all of the cells that are in the center of these masses undergo apoptosis and die, such that you create a hollow lumen. So we think that this process that we're seeing here is very similar to the process of lumen formation in vivo. And we found, as I'll show you, that if we put an oncogene in there that causes constitutive proliferation, if it only can cause hyperproliferation, all those cells die. And I'll show you that to you. So what we've done with this model is to introduce genes that have been implicated in breast cancer through a viral vector into the cells, into these cells, and then look at the effects of these genes on the architecture of the structure, on the proliferation properties, survival properties. And what we've done now is we've been able to mimic many of the architectural changes that you see in the breast, and now we're actually dissecting the cellular pathways that are responsible for this so that we can work with drug companies to develop molecules that will inhibit those processes. And I'll just show you a few examples. So we've looked at actually 289 different genes. So there's a group that's headed by Josh LeBaire that we've been collaborating with, and we're generating we're generating a library of 1,000 different genes that have been shown to be altered, either upregulated, downregulated, or mutated in breast tumors. And so far we've looked at 289 different genes. And I don't have time to show you what we found, but what we've been able to do is recapitulate many of these events that I told you about. So if we overexpress a protein called cyclin D, any of you ever heard of cyclin D? Okay, so cyclin D binds to the kinases that promote cell cycle progression during G1 and S, G2M phase. So if you, in 50% of human tumors, cyclin D is overexpressed. So if we just overexpress cyclin D in these cells, what we get is this hyperplasia. So we get structures that look similar to this. They're hollow. These are nuclear stains again. They're hollow, but as I'll show you, they keep proliferating, but they're hollow. There's no change in the architecture per se. If we overexpress a growth factor receptor called insulin-like growth factor, if we just overexpress it and incubate it with its ligand, either insulin or insulin-like growth factor, it causes constitutive proliferation, so these structures get really big, and as you can see here, they're filled. So the center of the lumen is just filled. And then it shows best by a phase contrast image. We found other genes which are able to cause invasive growth, but none of the genes we've looked at can take a normal mammary epithelial cell and make it invasive. We have to take these proliferative structures and add on top of it these other genes. And then we get a progression from being non-invasive to being invasive. So, it's not surprising that it would take more than one hit or one protein to convert a cell from this kind of normal ground state to being an invasive tumor, or show invasive properties. Let me just show you one of the genes, which is that's very nice that Hal gave such a good introduction to this. So one of the first genes, just almost as a proof of concept, that we put into our model system was the HER2 gene. And as Hal told you, HER2 is a growth factor receptor, and it regulates proliferation and survival in the normal gland and in other tissues. What he didn't mention is that there's one particular type of carcinoma in situ, and it's distinguished by its histology. So all of this one type of carcinoma in situ called comedo, C-O-M-E-D-O, has a necrotic core. So these are non-invasive. You can see the basement membrane around that. So it's been reported that 80 to 85 to 90% of comedo type carcinoma in situs have amplified HER2 neu. So it's something that people basically don't really pay much attention to because they're only following the invasive tumors. So the fact that such a high percentage of this one histological type of tumor has amplified ERB2 tells me that it's likely that HER2 is driving this the generation of this non-invasive carcinoma, and it's only when there's additional hits that this can become invasive, like you see with 30% of human breast tumors. So we wanted to see what kind of lesions would be generated if we express the HER2, if we overexpress the HER2 protein in our model. So these are the normal structures, and we actually were able to manipulate this gene so that we could turn it on with we could inducibly activate it. So we took structures which were perfectly normal, and then we activated HER2, which I won't go into how we did that. But what happens is these cells, which are growth arrested, they're non-proliferating, and they can't proliferate. You can add all the growth factor you want, they won't proliferate. If we turn on HER2, the cells proliferate like crazy, and they proliferate in all directions. So they proliferate within that monolayer, so they get a bit larger. They also proliferate to the outside, so they generate an additional spheroid-like unit, and then they proliferate to the inside such that they fill the luminal space. So if you did a cross-section through these, you would see that there was no hollow lumen anymore. So we can mimic this non-invasive carcinoma with by introducing this known breast tumor oncogene into these structures. So what we found, in fact, is that this is the ability to induce structures like this are not found with any oncogenes which cause constitutive proliferation. So this shows the ERB2. Actually, here's a cross section. You can see that most of these structures, not that one and that one, but they're filled with cells. And you don't see a hollow lumen. You see hollow spaces, but not, for the most part, they're not hollow. But if we, as I mentioned before, if we overexpress cyclin D, or another protein which I won't go into here, if you overexpress cyclin D, you actually cause hyperproliferation. But all of the cells that proliferate into the center, and they undergo cell death. So these cells are stained with a specific marker for apoptosis. So there's a family of proteases called caspases that are activated during programmed cell death. And this is an antibody that only recognizes activated caspase 3. And you can see that the cells that proliferate into the center die. So these cells' hyperproliferation does not appear to be sufficient in order to allow the filling of this luminal space. And it's something that we and most others hadn't thought about. We thought that those filled structures and tumors would just be filled because the cells are hyperproliferating. But our studies suggest that, in fact, you need not only a hyperproliferative signal, but also a signal that prevents the cell death. And this structure was stained with that same caspase 3 antibody. And you can see here in green, which is kind of showing up as aqua here, there's a few cells that are dying, but for the most part, ERB2 is preventing death of cells that are filling that cavity. So ERB2, which is a growth factor receptor that activates lots of cellular pathways, it can confer both a hyperproliferative state and an anti-apoptotic state. just driving the cell cycle with cyclin D causes hyperproliferation, but does not prevent the cell death. Now, one thing, and so without that, you get a hollow lumen, and the architecture of the structures don't really change. But what Jay Debneth, a postdoc in my lab, did said, well, what if I put an anti-apoptotic insult into these structures that have cyclin D? So he overexpressed a protein called BCL2, which is an anti-apoptotic protein, and it prevents apoptosis, and this is what you get. So the lumen fills up. So if you combine an anti-death signal and a proliferative signal, you can get a filled luminal space. So we imagine that during the evolution of tumors, some of them, like those which have amplified ERB2, that insult is going to be sufficient to cause the filling of the luminal space and a carcinoma in situ-like structure with a filled lumen. Whereas others may evolve by, say for instance, initial insult like activation of cyclin D or loss of RB, protein that regulates the cyclin-dependent kinases, plus, in addition, a second hit, which is anti-apoptotic. And one can imagine that the fate of these two different types of tumor cells is going to be different. That those with ERB2 may be able to progress to more serious invasive like lesions like the ERB2s do, whereas these won't because they only have, they've only been tickled in terms of their proliferation. They don't have this really, ERB2 is more like a neutron bomb. It hits so many intracellular pathways. Okay, so, I added this when I was a second version of it. So just to say, we've introduced a lot of different genes, and each one of the hyperproliferative genes causes a distinct architecture of the structures that we see. Like these two genes here not only cause hyperproliferation and filling of the luminal space, but it also breaks down the junctions. So this gene, when you activate this gene in the cells, you're much more likely to be able to generate an invasive structure later on because the cells are no longer tightly packed with each other. They're not held back by the cell-cell interactions. So basically you can generate these structures, and what we're doing now is trying to dissect the mechanism. There just shows you that. So we think that the spectrum of architectural changes that we see in our model reflect those in vivo. So a cure for cancer, the challenge, because of many alterations in breast cancer, the complexity of disease, it's difficult to find a single cure. Now, I don't want to rule out that there aren't going to be some therapies that are going to hit a lot of different types of tumors. And the kinds of therapies that are being worked on now that could potentially have much broader spectrum activity are the anti-angiogenesis strategies, which I talked about before. If you could prevent new blood vessels from coming into the tumors, once the tumor reaches a certain size, it's going to be starved of oxygen and nutrients. So if we could figure out a way to selectively block new blood vessel formation in tumors without affecting our normal blood vessels, that would be a really effective means of preventing more pan therapy for cancer. And I don't know if you've heard about this, but there's enzymes that maintain the ends of the chromosomes, telomerase, and once the chromosomes reach a certain size, when you replicate it, I shouldn't go into this, maybe. It's too late. No? It's okay? Okay. So normally, there's a process to replicate these ends, and in early stem cells, an enzyme complex called telomerase is able to replicate those ends. But in the progenitor cells that have a short which have a short replicative capacity, telomerase is shut off. And what happens is that you keep getting progressively shorter chromosomes because the telomerase isn't copying the ends of the chromosomes. And there's a certain point when the chromosomes reach a certain size that triggers an induction of cell death. And so if you take, say, breast epithelial cells and culture them in tissue culture, telomerase is off in those cells because there's just a few progenitor cells in there, and you get this crisis state, and the cells undergo cell death. So tumor cells have actually evolved a way to either turn on telomerase again so that you replicate those ends, or they figured out there's an alternate cellular pathway that allows replication of the ends of chromosomes. It's really not well characterized yet. But a tumor cell has to be able to replicate these ends, otherwise the cells will die. And so if you could, because a lot of tumor cells have upregulated telomerase, if you could inhibit the telomerase, the cells would kind of eventually just self-destruct, because the chromosomes would they would reach that point where they no longer can replicate, and you would induce apoptosis or just become non-proliferating. So that's another potential therapy, inhibitors of telomerase. And then obviously a lot of people are trying to induce immune responses to tumors, so that you could beef up the immune system and allow the immune system to be to escape those, or to recognize the tumor, because there's so many different ways in which a tumor can escape the immune response. So that's another kind of somewhat generalized but not entirely generalized strategy. But I think that it's very likely that we're going to have a combination of these kind of general approaches as well as these designer approaches, where you specifically target tumors based on their specific insults that are responsible for promoting proliferation and survival of those tumor cells. And the two designer drugs that I was going to tell you about are the HER2 antibody, which Hal told you about, so I don't need to tell you about that. So basically, a HER2 antibody binds to the receptor, it induces death of the cells by not totally clear mechanism. And then the other one is anti-estrogen therapy. So those tumors, many tumor cells are dependent on estrogen for proliferation and survival. And so if you use an antagonist that blocks estrogen from binding to its receptor, then you basically starve those cells of the signals that are required for proliferation and survival, and you eliminate the tumor. So in a way, anti-estrogen is probably one of the very first designer strategies. So for estrogen receptor positive cells, tamoxifen and other similar estrogen antagonists are really showing enormous clinical efficacy in preventing the proliferation of estrogen responsive tumors. So future drugs are going to require understanding the pathways that drive formation of different types of tumors. We want to identify the proteins that are altered and that play a primary role, not a secondary role, in the disease, and develop strategies to inhibit those. And initial successes in breast cancer and leukemias give us hope for the future. So we're clearly feeling much better about the prospect of eliminating cancer, even though it's a very complex disease. And I would just like to say here at the end, help. Get the students excited about what's happening in biology research. We still need to keep the pool of really talented students coming into the pipeline. Give them whatever chance you can to experience the thrill of discovery, no matter how trivial. Hopefully you can convince your principals to allow there to be some kind of science fair projects in the curriculum, so that the kids get a chance to get addicted to research. And clearly, we have a real problem now. The budget's not increasing, and we're spending money on things that a lot of us question. And the more you can contact congressmen to support continued interest increases in NIH and NCI budgets, the better. And if those aren't important enough, there's a really amazing statistic. So reducing cancer death by 20% would return $10 trillion to Americans. So we could significantly increase the amount of pool of money that would be available for things like education. And I just want to acknowledge, what? It won't go there. So I have a fabulous group of people to work with. I'm really privileged to have such a talented and dedicated group of students and postdocs and research assistants. We receive funding from several sources, and we're obviously very grateful for that as well. So thank you very much for listening.